Rolling between మన వెక్టర్ కాదు కదా అది నాకు డైరెక్ట్ ప్రతి స్టెప్ ఆపుకుంటానే చేసాం కదా ఒకటి క్లియర్ చేసుకుంటా
ఇప్పుడు ఎందులో వెళ్ళాలి కూడా సార్ ఏం తోచట్లేదు
ఎత్తి ఏం కాదు దాని గురించి
సార్ వచ్చాక అడుగుతారు కదా సార్
ఓకే వాట్ ఆర్ ద అదర్ restriction sites in that one?
Good afternoon, everybody. So we'll just start the session in 2 minutes. So today we'll discuss the questions which were covered in week 12 of our chapter, that is Introduction to Developmental Biology, and this is your last online session for the question per se. We'll have one more session tomorrow, that is on our scheduled class, same timing from 7 to 9 p.m. and there we'll try to revise whatever we have learned up till now. So this is the today we'll be covering the questions that were covered in the last week's class, that is the week 12's class, and we try to discuss the concepts while covering the questions as we do in our classes. And then subsequently for the last week, because after that I think you have your final examinations, so for the last week what we'll do is that we'll try to have at least 1 or 2 questions from each week's class. Okay, so when we'll have questions from each week's class, then we'll kind of revise all the things that we have learned up till now. Okay, so that is what we are going to do precisely for today's and for tomorrow's class. So I have taken this class today so that you have time for the revision also, and we can cover the syllabus as per our scheduled time. So that's why today's class has been scheduled, and tomorrow would be your normal class like we have. So shall we start the class now? Okay, so I'm going to start the class. So our first question is, which of the following can be explained by the reaction diffusion model? Let me just, sorry. Yeah. Which of the following can be explained by the reaction diffusion model? It's formation of tooth cups, spots on a cheetah, zebra fish stripes, tiger stripes, or eye color. Okay. This is A. Okay. B, C, D, and E. Sorry, my writing is very bad today because I don't have the pointer in my pen because I'm in my lab still. So please bear with me for that. So yeah, so your question is, which of the following can be explained by the reaction diffusion model? The options are A, formation of tooth cusps, B, spots on a cheetah, C, zebrafish, D, tiger stripes, and E, eye color. You have 2 minutes to think, and then we'll discuss the question. Okay.
Class one, the working isolated. And first class, first line, class one is isolate case. There, another master. It will not do it. It can't make classroom DNA practical in very classes.
1 minute more.
The classroom DNA makes it unless we are passionate about since we understand extent and class but no non-person value from the peer. Clear. Learning to work it and it's hard.
Okay. So our question was, which of the following can be explained by the reaction diffusion model? So first of all, we need to understand what is a reaction diffusion model, right? So reaction diffusion model, it was proposed by Alan Turing. So this model briefly explains how patterns like spots, stripes, and waves can spontaneously arise in biological systems through the interaction of diffusing chemical substances, which we call as morphogens. Okay, so basically it describes patterns like spots, stripes, or waves, how it can spontaneously arise in biological systems by the interaction of diffusing chemical substances, which are known as morphogens. So, while Turing reaction diffusion model was originally proposed to explain the pigmentation patterns like stripes and spots, later research has shown that similar mechanism can also underlie certain morphogenetic processes, including tooth cusp formation. So you understand? So initially, this Turing's reaction diffusion model, it was originally proposed to just explain the pigmentation patterns. Okay, the pigmentation patterns like spots or stripes. But then when eventually it was hmm, eventually it was shown that similar mechanisms are also present in morphogenetic processes such as tooth cusp formation. So, in particular, if we say during tooth development, what happens is that signaling molecules such as FGF, SHH, BMP, Wnt, they interact and diffuse through the developing tissue. Okay, and these interactions, it creates specially periodic patterns of gene expression that determine where cusp will be formed. And this patterning, it fits the mathematically framework of reaction diffusion dynamics, where local activation, for example, with the help of SHH, and lateral inhibition, for example, with the help of BMP, it establishes repetitive peaks corresponding to cusps. So, with this, can we say that the first option is correct? Yes. So now after this information, let us try to look at the options. First is formation of tooth cusps. So is it correct? Yes, because just now we have discussed that these interactions, for example, the interactions with the help of signaling molecules such as FGF, SHH, BMP. So these interactions, it creates spatially periodic patterns that determine where tooth cusp will be formed. So formation of tooth cusp is the correct answer. Then second option is spots on cheetah. So basically these are the different examples where we have to tell that where these examples they follow the reaction diffusion model or not. So for the formation of tooth cusps, we have just now learned it in detail. Okay. So now let us look at the other options. Other one is spots on a cheetah. Does it follow the reaction diffusion model? Does it follow? Yes, it do follow. So basically, reaction diffusion model, it can generate spot patterns similar to those seen in cheetah, leopards, and other animals. So yes, spots on a cheetah is the correct answer. The next option is zebrafish stripes. Does it explain the reaction diffusion model? Yes. So, the alternating light and dark stripes in zebrafish are a classic real world example of a reaction diffusion system which involves pigment cell interactions. Okay. Then tiger stripes. Is it also correct? Yes, it is also correct. So, tiger stripes can be modeled using reaction different diffusion models also. So up till now, formation of tooth cusp, it is correct. It is explained by the reaction diffusion model. Spots on a cheetah, it is also explained by reaction diffusion model. Zebrafish stripes, it is also explained by reaction diffusion model. Tiger stripes can also be explained by reaction diffusion model. And the last option is eye color. So can eye color be explained by reaction diffusion model? No. So eye color is basically determined genetically and biochemically and not by morphogen based spatial patterning. So, eye color cannot be described by the reaction diffusion model. Okay, so this I hope this question is clear to everybody. Let us move to our next question. Hmm. Choose the correct statements regarding mechanisms of evolutionary change. So, mutations responsible for differences between species majorly occur in protein coding sequences. Second, mutation in regulatory sequences are relatively dispensable. Third, mutations to create phenotypic variations occur in genes expressed during embryogenesis. And fourth, evolutionary relevant phenotypic variations among species result from mutations in genes expressed in the adult stage. Okay, so I'll give you 2 minutes to think and then I'll switch the screen. Think what can be the answer for this question. Just a minute. Hmm. Sorry. So you have 2 minutes to think. One more minute left. Okay, so let us discuss this question. So let us try to okay, so the first option is mutations responsible for differences between species majorly occur in protein coding sequences. So, is this correct or incorrect? It is incorrect. Why? Because most evolutionary differences do not come from protein coding changes. Okay, instead, they often arise from changes in regulatory sequences like enhancers, promoters that control when and where the genes are expressed. Okay, so protein coding sequences are usually highly conserved because changes there, it can disrupt the essential function. So here it says mutations responsible for differences between species majorly occur in protein coding sequences. So no, protein coding sequences are never touched. Okay. So basically, uh, as much as our biology tries to not to touch the protein coding sequences and give whatever mutations to the relatively lesser essential parts of the genome, so these protein coding sequences are conserved. So all these mechanisms the biology or the system has created in such a way so that you know we can adapt our system in such a way that less and less mutations are being given to the body. Fine. So second option is mutation in regulatory sequences are relatively dispensable. No, it is incorrect. Basically, mutations in regulatory sequences are indispensable because mutations or these sequences are crucial for evolution because they allow the changes in the gene expression without disrupting the protein function. So they are important, not dispensable. Then third option is mutation to create phenotypic variations occur in genes expressed during embryogenesis. Is this correct? Yes, this is correct. Genes expressed during embryonic development, it control the formation of body planes, organs, and overall morphology. So small changes here, it can lead to major phenotypic differences among species. Okay. For example, limb length, number of digit, facial shape, etc. So yes, mutations in developmental gene, they are important for evolutionary morphological differences. Okay. Because see, the morphology, it keeps on changing, okay, and because the morphology has changed, and that is the reason why evolution has occurred. So next is evolutionary relevant phenotypic variations among species result from mutation in genes expressed in adult stage. Is it correct or it is incorrect? No, it is incorrect. Why? Because while adult genes changes more often, uh, which affect the physiology or behavior, it does not affect the large scale body plan of the evolution. Okay. So adult genes can show variation, including significant morphological changes, but mostly it arises from developmental, that is embryonic gene expression differences. Okay. So I hope this question is clear to everybody. So, as I said, the correct answer is mutations to create phenotypic variation occur in genes expressed during embryogenesis. Okay. Mm. Hmm, fine. So, the next question is which of the following mechanisms are true regarding heterotropy? BMPs are responsible for interdigital apoptosis in bats. Germline antagonizes BMP in batwing. Dermally expressed FGF10 attracts rib precursor. BMPs are expressed interdigitally in all vertebrates. Mice deficient in BMPs will retain the interdigital webbing. You have 2 minutes to answer this question and then we'll switch the screen, okay? One more minute left. Okay, so let us discuss this question. Which of the following mechanisms are true regarding heterotopy? BMPs are responsible for interdigital apoptosis in bats. Is it true or false? It is true because in general vertebrate limb development, BMP signal cell death, that is apoptosis in the interdigital region. So in bats, BMP, they do induce apoptosis, but this is inhibit in certain regions to form the wing membrane. Okay. So yes, BMPs are responsible for interdigital apoptosis, the default pathway that gets suppressed where webbing is retained. Fine. The next is Germin antagonizes BMP in bat wing. Is it true or false? It is true because gremlin is a BMP antagonist. So in bat wings, it is expressed in the interdigit regions where webbing must be retained. Therefore, it inhibits the BMP pathway, preventing the apoptosis. So this heterotypic expression of gremlin, it contributes to the wing development. So this is correct. Then third is dermally expressed FGF10 attracts rib precursor. Is it true? Yes, it is a true statement. Why? Because this refers to snake embryogenesis. In normal vertebrates, what happens that FGF10 expression is restricted to limb forming regions. In snakes, FGF10 is expressed along the trunk dermis, attaching the rib precursors everywhere, which leads to ribs all along the body. So, this is a heterotypic change in FGF10 expression. Then, second last option is BMPs are expressed interdigitally in all vertebrates. Is it true or false? It is true. BMP expression in interdigital region, it is a conserved feature across the vertebrates. The degree of apoptosis, it depends on whether BMP are antagonized or not. So yes, BMPs are expressed interdigitally in all the vertebrates. Okay. Then mice deficient in BMTP will retain the interdigital vertebrates again, as said that this is a wrong statement. Okay. So see, you should understand what BMP and CAM4 control. Okay. CAM control. So BMP, it controls uh basically the proliferation. Okay. So saying that mice deficient in BMP will retain the interdigital webbing is misleading because the BMP family, it is redundant. So knocking out a single BMP often has little or complex effect. Okay. And complete loss of BMP signaling is often embryonic lethal. It causes wide developmental defects, not a simple syntactal phenotype is observed. So, in practice, conditional or local reduction of BMP signaling in the interdigital mesenchyme, it can cause webbing retention, but the blanket statement that mice deficient in BMP is too general. So, basically, you have to understand that it is a too generalized statement that has been used. Okay. So, I hope this question is clear to everybody. Now we'll move to the next question. So the accepted answers are BMPs are responsible for interdigital apoptosis in bats. Gremlin antagonizes BMP in bat wing. Dermally expressed FGF10 attracts rib precursor, and BMPs are expressed interdigitally in all the vertebrates. Okay, fine. Now, next question is match the following expression patterns with their corresponding beaks, width, and length. So, as you can see, you have a, graph where you have embryo A, embryo B, and embryo C. The blue bar represent BMP4 and the orange bar, it represents CAM. Okay. So, you have to match the following expression pattern with the corresponding beaks. So you can see you have a proper table which is given to you. Okay. So according to this table, you have to solve it. So you have the options A, B, C, D, E, F, where A option has this whole choice, that is A is narrow and long, B is wide and short, C is medium width and short. B option says that this embryo, so this is the option number. A B C D E F is the option number, and this A B and C are the embryo A, embryo B and embryo C, the three different graphs. So you have to uh, matlab identify that embryo A, embryo B, and embryo C according to the BMP4 and CAMP pathway. What can be inferred about the beak width and length? So width and length, width and length. So for embryo A, the beak is narrow and long. For embryo B, the beak is wide and short. For embryo C, the beak is medium width or short. So this is one whole option A, where it says A has this feature, B has this feature, C's beak has this feature. So like this, you have all the options. Okay. So I'll give you 3 minutes because it's a little lengthy question. So try to answer it, and then we'll discuss after 3 minutes. Okay, so try to first, you should know the relation between BMP4 and CAM. What is the relationship between BMP4 and CAM, and how BMP4 and, or even if not the relationship, how BMP4 and CAM control the beak length or beak width. So obviously from the question it can be said that BMP4 and CAMP, they control the beak width and length, but how they control, there is a mechanism behind it that you should know. If you know the mechanism, then according to the, see, there it is a fold difference. Fold difference is given to you. So according to the fold difference, you can tell from the given options that whether it is which of the statements or the combinations is correct. 2 more minutes left. See the options, try to match with the options, then only you will be able to solve it. Okay, so let us discuss this question now. Fine. Okay, so let us try to understand this question. So step one, we'll try to understand that what BMP and CAM controls. Okay, so BMP, that is bone morphogenetic protein, it controls beak depth and width. So higher the BMP4, broader and deeper the beak. And CAM, what is CAM? CAM is cal module. It controls the beak length. So higher CAM, that means longer beak. So understand this. Higher BMP4 means broader and deeper beak. Okay. Higher BMP4, broader and deeper beak. And higher CAM, that is cal module, that means there is long beak, longer beak. Okay. So if high BMP4, then what is there? Beak is wide, but it can be short. It depends on cam. High cam, so beak would be long, but it can be narrow. We don't know because we don't know about cam. But if both are high, then it is wide and long. If both are low, then narrow and short. So this is the gist. BMP, it controls the width, and cam, it controls the length, and both are positively related to the length, to the width and the length. So now let us try to understand the graph. Okay, so let us look at embryo here. A. BMP is low and CAM is high. So this means CAM is very high. Okay. Then what we can say, it is narrow because BMP is low, and long because BMP controls the width and your CAM controls the length. So BMP levels are less, that means it would be narrow and not broad. But because CAM levels are very high, so it would be long. Okay, so narrow and long. So narrow and long, this is narrow and long. None of the options have narrow and long. So from this only we can get to see that A can be the answer. Then second is BMP levels are very high, that means it is kind of wide, but cam levels are very low, so it is length is short, so wide and short. Okay, then BMP levels are not very high, not very low, it is in between somewhere. So see, because how we can say it is somewhere in between, you can see here it is 6, which is very high, and here it is approximately 3, it has gone maximum up till 6. Here it is approximately 3, so we can say it is somewhere in between. So it is medium, not very broad, not very narrow. And here it is less only, so that means length is short. So medium, width and short. So from this we can say that option A is the correct answer. Okay. So I hope this question is clear to everybody. Let us now move to the next question. But before that, see, this is what happens. Hmm. Fine. So the accepted answer is A. So the next question is, if A is heterochrony and B is heterometry, which statements are correct? Okay, if A is heterochrony and B is heterometry, which statements are correct? Okay, statements are B is equals to SHH overexpression causing down regulation of PAC6. B, SHH overexpression mediated degeneration of eyes. A, FGF8 mediated elongation of the middle digit of dolphin flipper. And the last option is A is equals to SHH mediated production of digits in a lizard. This is your question. So for solving this question, you must know the meaning of heterochrony and the meaning of heterometry. Okay. So if you don't know the meaning of heterochrony and heterometry, it is little difficult for you to solve this question. But if you know what is the meaning of heterochrony and heterometry, then it is to try to solve this question. 2 minutes to solve this question, and then we'll discuss. One more minute left. Okay, so let us discuss this question. So first of all, we'll try to understand what is the meaning of heterochrony and heterometry. Okay. So heterochrony means change in timing or duration of gene expression. Okay. What is the meaning of heterochrony? Change in the timing or duration of gene expression. And heterometry means change in the amount, that is the level of gene expression. That is heterometry. So now let us try to look at the options. The first option is, if B is equals to SHH, then overexpression causes down regulation of PAX6. So is it true? Yes, it is heterometry. Overexpression is equals to change in the level of SHH, which is directly a heterometric change. And this altered level, it leads to PAX6 down regulation, which is consistent with heterometry. Then if B is equals to SHH overexpression mediated degeneration of eyes. Is it true? It is also true. It explains again heterometry. So the eye degeneration, it results from increased SHH expression levels along the midline, and that's a quantitative change. So again, it is what? It is heterometry.
And they get 3 micrograms a month.
Then FGF8 mediated elongation of the middle digit of dolphin flipper. What is it? It is heterochrony. So again, it is true. Because the dolphin flipper's elongated digit, it results from prolonged expression of FGF8 in the apical ectoderm ridge. And that's a change in the duration or timing, which is a perfect example of heterochrony. Then A is equals to SHH mediated production of digits in a lizard. Is it true? Yes. So in this case, the timing and duration of SHH expression during limb development determine the number and identity of the digits formed. and species specific digit variation, it often involves heterochronic shifts in acetate expression, not just the spatial or quantitative change. So this is also true. So ultimately we can say that all the options are correct. So first two first two options are the examples of heterometry, because both are related to the change in the amount or the level of gene expression, that is a quantitative change. And the last two are the examples of heterochrony, because both of them are related to the change in timing or the duration of gene expression. Okay. Okay, now comes the question. The following gene, just a minute. Okay, so the question is, the following gene is divided into six modules A to F. If a mutation occurs simultaneously in two modules, which pairs are both likely to cause a heterotypy? So now we know what is heterotypy, right? Heterotypy, no, we have discussed heterometry and heterochrony. Okay, so we'll discuss it now. So the following gene is divided into six modules A to F. If a mutation occurs simultaneously in two modules, which pair are both likely to cause heterotypy? Your options are uh, A to F, A to B, B to E, C to E, C to F, and D to F. D and F. Just ignore this thing. This is nothing. Just see the question. I'll give you 2 minutes to think, and then we'll switch the screen and we'll discuss it. Okay.
No basement, no. Really not counting something.
So first you should know, for this question, to solve this question, you should know the transcription initiation region, the translation initiation region, the terminal exon or terminal coding region. So these things you should know. Shall we discuss the question? Let us discuss the question now. So let us try. So B is the transcription initiation or the 5' region. Okay. So changes here will alter where and when the gene is transcribed, and that directly produces a heterotopy. Okay. C is the translational translation initiation region, the N-terminal coding region. So mutations here, it can alter the N-terminal signal peptide or other targeting motifs that send the protein to a new cellular or extracellular location. So that is a change in spatial deployment of the protein, which is heterotopy. And E portion, the E portion, this is the terminal exon, including stop codon, which is the C-terminal coding region. Okay. And C-terminal sequences, they often contain localization or retention signals. For example, membrane anchors organally targeting localization motifs in the protein or signals affecting mRNA localization. And mutation in E, it can therefore retarget the protein spatially. So what we can say overall, B plus E is equals to change where the gene is expressed, okay, and change where the protein produced is localized. The where the gene is expressed, that is your B, and where the protein which is produced is localized, which is E. So which is heterotopy, and C plus E is the change in the N-terminal and the C-terminal targeting signals of the protein. That means relocalization of the protein product, which is again heterotopy. Other combinations are not directly tied to changing the spatial targeting, and therefore the accepted answers in this case would be B and E and C and E. Okay, fine. Now next question is, select all correct statements regarding developmental mechanisms and constraints. Developmental constraints restrict the number of phenotypes only if an embryo develops in a restricted environment. Limb bud grafted onto the trunk develops into a normal limb. This is enabled by the dissociation of modules. All mechanisms of evolutionary change only exploit the modularity of enhancers, and mechanisms of evolutionary change aid only if intraspecific variation and selection. These are your options. You have 2 minutes and then we'll switch the screen. Okay, so let us discuss this question. The first option is, developmental constraints restrict the number of phenotypes only if an embryo develops in a restrict. Just a minute. Right. Hmm. Hmm. So developmental constraints arise. The option is a developmental constraint restricts the number of phenotypes only if an embryo develops in a restrictive environment. So this is an incorrect statement because developmental constraints they arise from the inherent structure and interactions of developmental systems, not just the environmental conditions. Okay. So they can limit the phenotypic variations even in a perfectly normal environment because some forms simply cannot be produced by the developmental program. So this is false because constraints are intrinsic, not only environmental. Okay. Then limb bud grafted onto the trunk develops into a normal limb. This is enabled by the dissociation of modules. It is correct because in developmental biology, modules can function semi-independently, a property which is called as modularity. So because of this dissociation of developmental modules, a grafted limb bud can still follow its own intrinsic developmental program and form a normal limb even in a new location. So it is true, that is, which demonstrates modular and autonomous development. Then all mechanisms of evolutionary change only exploit the modularity of enhancers. This is again incorrect because while enhancer modularity, it is a key mechanism which allows the independent control of gene expression, evolutionary charge can also happen because of many other reasons. Okay. And what can be those reasons? It can be changes in the coding sequences, which is known as heterotypy, timing, which is known as heterochrony, level, which is known as heterometry, location, which is known as heterotropy, etc. So it is false. Then mechanisms of evolutionary change aid only intraspecific variation and selection. It is incorrect. So
Hmm?
this is incorrect because mechanism like heterochrony, heterotopy, heterometry, heterotropy, they operate both within the species and between the species. So both within the species known as microevolution and between the species is known as macroevolution. Okay, so it is incorrect. Then limb bud grafted onto the trunk develop into a normal limb. This is enabled by the dissociation of molecules. So what is this? This is
నొక్కలేదు అది ప్రెస్ చేయాలి అది ఎరుగుతుంది చూడు
correct. So why it is correct? So see,
ప్రెస్ చేయాలి
physical constraint
అవును సెట్ అయితే వేరే
means
కలవచ్చు
limitation due to the physical laws or mechanical properties of tissues. For example, size, growth, gravity, diffusion limits. These are the examples of physical constraints. And morphological, morphogenetic constraints. These are the constraints that happens due to developmental programs or morphogenetic pathways. For examples, sequential patterning, fixed morphogen, and gradients. And phyletic constraints are the constraints due to evolutionary history and shear ancestry. And what are the examples of phyletic constraints? Examples are conserved developmental stages, body plans. So, with this summary in mind, the only correct statements is the limb bud grafted onto the trunk develops into a normal limb, which is enabled by dissociation of modules. Okay. Next question is we have to select the correctly matched pairs. You have physical constraint, morphogenetic constraints, and phyletic constraints. We have just discussed this also. Shall we discuss this question now? Okay, so let us discuss these questions. So the first accepted set for this question is A1, B4, and C3. So, and the second accepted set for this question is A2, B7, and C5. And what is physical constraint, what is morphogenetic constraint, and what is phyletic constraint, we have already discussed. Okay. Okay. The next question is select the correct statements regarding epigenetic inheritance system. It allows environmentally induced changes to be inherited. Epialleles are variants of DNA sequences. Epiallelic modification occur in the germline, and hypermethylation of a gene will have the same effect as a loss of function gene mutation. Okay, so let us try to understand this question. First option is it allows environmentally induced changes to be inherited. So, what we have to talk about, we're talking about the epigenetic inheritance system. So, it allows environmental induced changes to be inherited. See, this epigenetic, it does means that environmental changes are inherited, but is it always the case? No, it is not always the case because while epigenetic mechanism, it can respond to the environmental inputs. Most environmentally induced epigenetic changes are not stably inherited across the generations. Okay. So whatever environmental changes are happening, it happens in an individual and it is mostly restricted to that individual. These are not inherited across generations. Fine. So germline reprogramming, it usually erases the epigenetic marks. So only a few exceptional cases are there. Otherwise, mostly what happens that these changes are not stably inherited. So it's not considered a general rule that environmentally induced changes will be inherited. So it is a wrong statement. Second is epialleles are variants of DNA sequences. Is it true or false? It is false. Why? Because epialleles differing epigenetic state and not the DNA sequences. Okay. So it is straightforwardly incorrect. There's nothing else to explain about. Then epiallelic modifications occur in the germline. Is it correct or not? It is correct. Because epigenetic mark, if you want any epigenetic mark that has to be inherited, then it should occur or it should persist in the germline cells. Okay. Otherwise, it is erased during fertilization or embryogenesis. Then, hypermethylation of a gene will have the same effect as loss of function gene mutation. It is correct or not? It is correct. Because hypermethylation of promoter region, it silences transcription. So there is no functional protein that is present and functionally equivalent to a loss of function mutation, which is even though the DNA sequence is intact. So this is correct. Fine. Now let us come to the last question. Just a minute. Okay. The last question is which of the following statements are correct in the context of genetic assimilation? First, the selection of dark colored moths, as described in industrial melanism, is a prime example of genetic assimilation. Second, genetic assimilation can cause intraspecific differences in phenotype. It enables adaptation. It requires an initial mutagenesis to develop the phenotype, or it is enabled by the developmental plasticity of structures rather than the induction of new structures. Okay. You have 2 to 3 minutes to answer this. Okay. So let us discuss the question. First option is the selection of dark colored moths as described in industrial melanism is a prime example of genetic assimilation. Is it true? Yes, it is a true statement. C. Although industrial melanism is classically described as a case of natural selection on existing genetic variation, in a broader interpretation, it can be used as a case where environmentally induced pressure, for example, pollution, it led to the fixation of a phenotype which is dark coloration. Okay. And this is seen as conceptually parallel to the genetic assimilation, that is, environment phenotype genetic fixation. So, while not a strict example, you can say it is still analogous to what we have learned. So therefore, it can be accepted. Then genetic assimilation can cause intra-specific differences in phenotype. Is it correct? Yes, it is a correct statement. See, population in different environments, it may fix different phenotypes that were initially environmentally induced. Okay. So within a species, distinct forms may emerge via genetic assimilation. Then it enables adaptation. Yes, it is also correct. The entire process, environmentally induced phenotype, heritable fixation, everything is adaptive, as it allows the organism to stabilize useful traits that were once flexible responses. So yes, it enables adaptation. Then it requires an initial mutagenesis to develop the phenotype. Yes, it is also correct. So the genetic fixation phase, it involves some mutational changes that makes the plastic response constitutive. That means that makes the plastic response permanent. So if the question is it requires later mutagenesis to stabilize the phenotype, then it is accepted correct. Okay, so this is correct statement only. Then it is enabled by the developmental plasticity of structures rather than the induction of new structures. Is it correct or not? Yes, it is correct. So see, the core concept is that genetic assimilation, it works by stabilizing the pre-existing plasticity. Okay. The genetic assimilation, it works by stabilizing the pre-existing plasticity and not inventing entirely new organs or structures. So it is the correct statement. So have you understood this? Okay, so the correct statements are: the selection of dark colored moths as expressed in industrial melanism is a prime example of genetic assimilation. Then second, genetic assimilation can cause intraspecific differences in phenotype. It enables adaptation. It requires an initial mutagenesis to develop the phenotype, and it is enabled by the developmental plasticity of structures rather than the induction of new structures. So all the options are correct. So is this question clear to everybody? Let me know if this is clear or not. Hmm. So fine. I hope that you were able to understand the questions. And tomorrow again we'll meet at the same time, 7 to 9 p.m. And I'll take one question from each class. So we'll have 12 questions. Okay. So we'll try to discuss 12 questions. So I'll take one question from each class, and then we'll discuss it. So in that way we'll revise also. So we'll do some revision tomorrow. So this is it for the day. Bye bye everybody. We'll meet next week. No, sorry, tomorrow. And then you have your exams, right? So all the best for the exams. Do well. Bye.